Welcome to the Deep Dive, where we tackle complex topics and distill the most crucial insights directly to you. Today, we're taking a deep dive into the truly astounding metabolism of the nervous system.
It really is something else.
Think about it. Your brain, though it's only about, what, 2% of your body weight?
About that, yeah.
It demands a phenomenal 20% of your body's oxygen and just a huge amount of glucose. This isn't just an interesting fact. It means the brain's biochemical processes are absolutely central to, well, every single thought, feeling, and movement you experience.
Precisely. And for this deep dive, our mission really is to offer you a comprehensive sort of step-by-step understanding of the brain's unique internal workings.
Okay.
We'll explore its specialized cellular architecture, the incredible protective systems in place, how it communicates through this complex language of neurotransmitters, and what happens when these vital systems face challenges.
So kind of a shortcut to understanding the brain's biochemistry.
Exactly. Consider this your shortcut to grasping the brain's profound biochemical demands and functions.
Okay, let's unpack this then. When we visualize the brain, we often think of neurons, those famous signal transmitters. But what's truly fascinating, I think, is that neurons are just one part of a much larger dynamic ecosystem. Who are the other key players, and what roles do they perform in keeping our nervous system, not just alive, but thriving?
It's so true. Neurons are, you could say, the principal actors, but they rely heavily on a sophisticated supporting cast we call neuroglial cells.
Right, the corellia.
Exactly. First, the neurons themselves. They're specialized for transmitting signals, with dendrites to receive information and axons to send it out, all connecting at synapses. A crucial insight about neurons is that they are terminally differentiated.
Meaning they don't really replace themselves easily?
Pretty much. This means, unlike many other cells in your body that can regenerate, neurons largely mature into their final form without the ability to easily divide or replace themselves. This highlights just how precious each neuron is. Significant damage often leads to programmed cell death or apoptosis.
Okay, got it. So what about that support crew, the neuroglia?
Right, the amazing support crew. In the central nervous system, or CNS, we have astrocytes. These star-shaped cells are more than just physical support, they're active caretakers.
What do they do specifically?
Well, they provide vital nutrients. They even convert glucose into lactate to feed hungry neurons sometimes. They guide neuronal migration during development and meticulously regulate the ionic environment around neurons. They're constantly shaping the neuronal landscape.
Wow, busy cells.
Very busy. Then there are oligodendrocytes, also in the CNS. These are the unsung heroes of speed. They form the myelin sheath, that fatty, protein-rich covering that insulates axons.
Like insulation on a wire.
Exactly like that. This insulation dramatically speeds up the electrical signals. What's truly efficient is that a single oligodendrocyte can myelinate.
Wait, multiple axons?
Yeah, up to 40 different axons. It's incredibly efficient.
Okay.
Now, in the peripheral nervous system, or PNS, we find Schwann cells. They perform a similar myelination job, but they're a bit more focused, typically wrapping around only one axon.
Just one.
Just one. They also play a critical role in clearing cellular debris and significantly assist in repairing damaged peripheral axons, which is something oligodendrocytes don't do as well.
Interesting difference. Okay, who else?
Microglial cells. Think of these as the brain's resident immune cells. They're constantly on patrol, acting like tiny macrophages, destroying invading microorganisms and tidying up any cellular debris.
The cleanup crew.
Basically, yes. And finally, ependymal cells. These ciliated cells line the fluid-filled cavities of your brain and spinal cord. They secrete and circulate cerebrospinal fluid, or CSF.
The CSF, right. Shock absorber and waste removal.
Exactly. It acts as a shock absorber and helps remove metabolic waste. What's particularly intriguing is their potential as neural stem cells, which hints at, future regenerative therapies, perhaps.
That's a comprehensive network of support. And with all these delicate processes and crucial cells, I'm particularly struck by how the brain protects itself. How does it keep harmful stuff out while still managing to get all the specific nutrients it needs? It sounds like an incredibly precise and tightly regulated system.
It absolutely is, and this precision is largely thanks to the blood-brain barrier, or BBB.
The famous BBB.
Indeed. And this isn't just a simple filter, it's a sophisticated gatekeeper. The capillaries in your brain are unique because their endothelial cells are stitched together by extremely tight junctions.
Tighter than elsewhere in the body.
Much tighter. Unlike the leaky capillaries elsewhere, this prevents polar molecules from simply slipping through. They also lack the usual windows or fenestrations and the bulk transport of vesicles called transpinocytosis that you see in other tissues.
So it's physically sealed off, essentially.
To a large extent, yes. But the BBB's defenses go beyond physical structure. It's also an enzymatic barrier. The endothelial cells contain drug-metabolizing enzymes, much like your liver, that can neutralize potentially harmful chemicals.
Oh, interesting.
Plus, they actively pump out hydrophobic molecules that try to sneak in, using special efflux pumps called P-glycoproteins. And it's not just the endothelial cells, even the foot processes of those astrocyte cells we just discussed contribute to this multi-layered protection.
So the astrocytes are involved here too.
They definitely play a role. Now, despite all this strict control, the brain still needs its vital supplies. Nonpolar substances like many drugs, oxygen, carbon dioxide, and water can diffuse passively across. Pretty easily, in fact.
Okay, the small nonpolar stuff gets through.
Right. But for most other molecules, specific transport mechanisms are essential. For instance, glucose, the brain's primary fuel.
The main one.
The main one, absolutely. It uses facilitated diffusion via GLUT1 transporters on the endothelial cells and then GLUT3 to get into the neurons themselves. Different transporters for different steps.
GLUT1 at the barrier, GLUT3 into the neuron. Got it.
Precisely. There are also dedicated systems for monocarboxylic acids, such as lactate, and critically ketone bodies like acetoacetate and hydroxybutyrate.
Ketone bodies. Important during fasting, right?
Exactly. These become essential fuels during prolonged starvation, and their transport system actually gets upregulated when you need them most. Then you have large neutral amino acids, or LNAs, like phenylalanine, leucine, and tryptophan. They share a single transporter.
They share, so they compete.
They compete for entry, a critical detail. Vitamins also have their own specific transporters. For larger proteins like insulin and transferrin, the brain uses receptor-mediated transcytosis.
Receptor-mediated, like docking and being carried across.
Wonderful like that, yes. Imagine these proteins binding to a specific docking station on the barrier and then being ferried across in a small cellular package.
That's a truly intricate system of selective transport. Can you walk us through a clinical scenario where one of these specialized systems malfunctions? What's the real-world impact?
Absolutely. One striking example is GLUT1 deficiency syndrome.
GLUT1, the glucose transporter at the barrier.
That's the one. Here, the GLUT1 transporters are impaired, leading to dangerously low glucose levels in the cerebrospinal fluid, a condition called hypoglycemia, even though blood glucose levels might be normal.
So the brain is starving for glucose, even if there's plenty in the blood.
Exactly. Patients experience seizures, developmental delays, and motor disorders because their brains are simply starved of fuel. The ingenuity of treatment lies in a ketogenic diet.
The ketones again.
Right. This diet prompts the body to produce ketone bodies. These can then bypass the faulty GLUT1 system using that separate, upregulated transport system for monocarboxylic acids we mentioned, providing the brain with an alternative fuel.
Clever workaround. Any others?
Another important example tied to that LNAA transporter competition.
The shared one for amino acids.
Yes. That's relevant in phenylketonuria, or PKU. In untreated PKU, excessively high phenylalanine in the blood overwhelms the shared LNAA carrier. This competitive inhibition prevents other essential amino acids like tryptophan and tyrosine from reaching the brain.
So too much of one blocks the others out.
Precisely, which can lead to severe neurological damage and mental retardation if not caught early and managed with diet. The insight here is the delicate balance of transporters and how an excess of one substrate can starve the brain of others despite overall availability.
That clearly shows the profound impact of even tiny disruptions. So what does all this mean for how our brain actually communicates? We've talked about the cells and the barriers, but the real language of the brain is spoken through neurotransmitters. Let's dive into how these fascinating messengers are made, released, and controlled.
Indeed, neurotransmitters are the brain's language, facilitating every thought and action. Most of these small nitrogen-containing neurotransmitters share fundamental characteristics. They're typically synthesized right in the presynaptic terminal, the sending part of the neuron.
Made on demand, basically.
Often, yes, or stored ready to go. They're made from basic building blocks like amino acids or intermediates from glucose metabolism like glycolysis or the TCA cycle. Their synthesis rate is remarkably dynamic, adjusting to how frequently a neuron fires.
So activity influences production.
Directly. Once made, they're packaged into tiny storage vesicles, a process requiring ATP, energy.
Energy intensive.
Very. And held there until an electrical signal arrives. This signal triggers a rapid influx of calcium ions, causing the vesicles to fuse with the cell membrane and release their chemical cargo into the synaptic cleft.
And they need to be cleared out quickly, I assume.
Absolutely. Their action is swiftly terminated to ensure precise, rapid signaling. This can happen through reuptake back into the presynaptic terminal, absorption by nearby glial cells like astrocytes.
Glia again.
Or just diffusion away from the synapse, or by enzymatic inactivation being broken down by specific enzymes. This intricate dance of synthesis, release, and termination is central to the brain's incredible processing speed.
That's an elegant system. Let's zoom in on some of the, let's call them the big three neurotransmitters: dopamine, norepinephrine, and epinephrine. Where do these crucial molecules originate, and how does the brain manage them?
Okay, these three are all part of the catecholamine family, and they illustrate the brain's reliance on specific dietary precursors and enzymatic pathways. They all begin with the amino acid L-tyrosine.
Which we get from diet or make from phenylalanine, right?
Exactly. The pathway is a biochemical assembly line. First, tyrosine hydroxylase, requiring a critical cofactor called BH4, converts tyrosine into L-dopa. This is the weight-limiting step, the bottleneck of the whole process.
BH4, tetrahydropirin.
Okay. Then, L-DOA decarboxylase, which needs vitamin B6, turns L-DTOP into dopamine. If a neuron is dopaminergic, it stops here.
So dopamine neurons finish at dopamine. Makes sense.
Right, but if norepinephrine is needed, dopamine mahydroxylase, located inside the vesicles and requiring vitamin C and copper, steps in to convert dopamine.
Inside the storage vesicles. That's interesting.
Yes, it happens right there. And for epinephrine, the enzyme phenylethylamine N-methyltransferase adds a methyl group, a reaction that depends on adenosylmethionine, or SAM.
SAM, the methyl donor.
The universal methyl donor, which itself relies on adequate vitamin B12 and folate for its regeneration. The interconnectedness of these pathways with our nutritional intake is truly profound. You need the tyrosine, the B6, the vitamin C, the copper, B12, folate.
A lot of nutrients involved.
Absolutely. For storage and release, dopamine is actively taken into vesicles by VMAT2, a transporter linked to a proton pump. Inside, catecholamines are concentrated alongside ATP and other proteins called chromogranins.
Chromogranins.
Yes, and their elevated levels in circulation can actually be a marker for certain neuroendocrine tumors, like pheochromocytomas, that overproduce these. When a nerve impulse arrives, calcium influx triggers their release into the synapse.
And inactivation, reuptake or breakdown.
Both. Their action is terminated primarily by reuptake back into the presynaptic terminal or by enzymatic degradation. Two key enzymes here are monoamine oxidase, or MAO, found on mitochondrial membranes.
MAO inhibitors are used as antidepressants, right?
They are, exactly. MAO oxidizes the amino group and has forms like MAOA for norepinephrine and serotonin, and MAOB mainly for other amines. And the second enzyme is catechol O-methyltransferase, or COMT.
COMT?
COMT methylates a hydroxyl group, also relying on SAM, and thus B12 and folate. This entire system shows just how deeply cellular energy and specific nutrients are entwined with brain function. You even see specific breakdown products like homovanillamandelic acid, HVA, from dopamine, which is decreased in Parkinson's disease.
That's a powerful illustration of biochemical precision, and it brings us directly to a striking clinical example you mentioned in the outline. Katie C., can you explain her alarming symptoms through the lens of this catecholamine pathway?
Right, Katie C. She presented with a classic constellation of symptoms: palpitations, pounding headaches, profuse sweating, and dangerously severe hypertension.
Sounds awful.
It is. These are hallmarks of a pheochromocytoma, a tumor, typically in the adrenal medulla, that secretes excessive amounts of norepinephrine and epinephrine.
So just dumping catecholamines into the system unregulated.
Exactly. This throws the body into a hyperadrenergic state, essentially an extreme overdrive of the sympathetic nervous system. The diagnostic insight came from elevated levels of metanephrine, a methylated breakdown product of epinephrine, in her blood and urine, confirming the unregulated overproduction.
And treatment.
Treatment focuses on stabilizing the patient first with alpha and beta blockers to control the effects of the excess catecholamines, then followed by surgical removal of the tumor. It's a clear example of how unchecked synthesis of these vital neurotransmitters can wreak havoc.
That's a stark reminder of the delicate balance. Moving to another critical player, let's talk about serotonin.
Serotonin metabolism shares a similar elegance. It's synthesized from the amino acid tryptophan.
Another essential amino acid from diet.
Yes. The pathway involves tryptophan hydroxylase, again requiring BH4, followed by a decarboxylation step needing vitamin B6.
Same cofactors pattern again, BH4 and B6.
You see these patterns repeating. Like catecholamines, serotonin's action is primarily terminated by MAO, specifically MAOA.
Which links back to those MAO inhibitors.
Exactly. The importance of MAO in regulating serotonin levels is why MAO inhibitors was some of the earliest antidepressants. However, these drugs came with the infamous cheese effect.
Ah, yes, the tyramine issue.
Right. Dietary tyramine found in aged cheeses, cured meats, etc. is usually inactivated by MAOA in the gut and liver. With MAOIs blocking that, tyramine would build up and cause dangerous hypertensive crises by displacing norepinephrine from nerve terminals.
A major side effect.
Huge. This brings us to Evan A.'s experience with weight loss drugs. He was prescribed Redux, which worked by both increasing serotonin secretion and blocking its reuptake.
So a double whammy on serotonin levels.
Pretty much. In contrast, drugs like Prozac, a selective serotonin reuptake inhibitor, or SSRI, only block reuptake. While both increase serotonin signaling, their distinct mechanisms led to different outcomes. Redux was eventually pulled due to toxicity, including heart valve issues.
So how you boost serotonin matters.
It highlights the crucial difference in how we modulate these pathways and the potential for unintended consequences. It's a key insight into how tweaking even slightly different points in a biochemical cascade can have major clinical implications. Also, just as a side note, melatonin, the sleep hormone, is synthesized from tryptophan too, mainly in the pineal gland, following a similar initial path as serotonin.
Interesting connection. What about histamine and acetylcholine? How do these fit into the brain's biochemical story?
Histamine in the brain, also an excitatory neurotransmitter, is produced from histidine in a single step by histidine decarboxylase, which needs vitamin B6 again.
B6 again. Very important vitamin for neurotransmitters.
Extremely. Histamine is stored in vesicles, released, and then largely inactivated by methylation using SAM again and subsequent oxidation, often by MAOB rather than reuptake being the primary route.
So methylation and MAOB, not reuptake.
Primarily. Clinically, you know histamine from allergic responses. The fact that non-drowsy antihistamines are modified not to cross the blood-brain barrier is a perfect example of the BBB's selective function and how we manipulate it for specific therapeutic effects. They block histamine peripherally, but don't cause drowsiness by acting centrally.
Clever drug design based on the BBB. Okay, and acetylcholine.
Then there's acetylcholine, or ASHI. It's synthesized from acetyl-CoA and choline. The SO group comes mainly from glucose oxidation via pyruvate dehydrogenase, a process that absolutely requires vitamin B1.
Thiamine B1, crucial for glucose metabolism.
And therefore crucial for ASHIC synthesis. Choline comes from our diet or from membrane lipids, and its de novo synthesis involves 3 crucial methylation steps using SAM.
SAM again, so B12 and folate are important here too?
Critically important for regenerating SAM. This dependence helps explain why deficiencies in thiamine, B12, or folate can profoundly impact neurological function, partly through effects on ASEEC synthesis or choline availability. Acetylcholine's action is swiftly terminated by acetylcholinesterase, or ACE.
And that enzyme is targeted by nerve agents, right?
Exactly. Neurotoxins like sarin gas inhibit ACE, leading to a buildup of ACE and continuous nerve activation, which is often lethal.
A vital enzyme.
Yeah.
And finally, let's look at the brain's major excitatory and inhibitory players.
Absolutely essential. Glutamate is the primary movement of synapses.
The main go signal?
Pretty much. It's largely synthesized from glucose via the TCA cycle intermediate iketoglutarate using glutamate dehydrogenase or transamination reactions, or it can be made from glutamine supplied by glial cells. Glutamate is removed from the synapse very efficiently by high-affinity uptake systems in both neurons and glial cells.
Needs to be cleared quickly to prevent overexcitation.
Critically important. Its counterpart is GABA, or aminobutyric acid, the major inhibitory neurotransmitter, the main stop signal.
Balance between glutamate and GABA is key.
Absolutely. GABA is synthesized directly from glutamate by glutamic acid decarboxylase, or GD, which again needs vitamin B6.
B6 involved in making the main inhibitory transmitter from the main excitatory one.
Wow. It's a beautiful link. These two are interconnected through the GABA shunt, a metabolic loop, and the vital glial neuronal glutamine cycle. Here's how that works. Glial cells take up glutamate from the synapse, convert it to glutamine.
Which is less toxic.
Less directly excitatory, yes.
Yeah.
This glutamine then travels to neurons to be converted back to glutamate by an enzyme called glutaminase. This cycle ensures glutamate availability for both signaling and GABA synthesis, demonstrating a remarkable intercellular metabolic partnership. Critically, glial cells cannot make GABA themselves. They lack the GAD enzyme.
So neurons make GABA, glia help recycle a precursor.
That's the gist of it. Clinically, drugs like tiagabine, which inhibit GABA reuptake, are used to treat epilepsy by prolonging GABA's inhibitory effect, a clear example of manipulating neurotransmitter half-life.
Makes sense. Anything else in this category?
We also briefly note aspartate, another excitatory amino acid made from oxaloacetate, and glycine, an inhibitory one particularly important in the spinal cord, synthesized from serine and requiring folic acid for that conversion.
Folic acid for glycine.
Yes. And finally, there's nitric oxide, or NO.
A gas as a neurotransmitter.
Exactly. It's synthesized from arginine by nitric oxide synthase. NO acts as a biological messenger, causing vasodilation but also influencing neurotransmission. Because it's a gas, it diffuses freely across membranes and can even act as a retrograde messenger.
Meaning it goes backward.
Yes, flowing backward from the postsynaptic neuron to influence neurotransmitter release from the presynaptic terminal, a very different kind of signaling molecule.
That's an immense amount of detail on how the brain's internal communication works. What happens when these incredibly critical fuel and oxygen supplies are disrupted, or these intricate metabolic pathways begin to, well, falter?
This leads us into the serious realm of encephalopathies and neuropathies. The core insight here is the brain's absolute non-negotiable dependence on a constant supply of glucose and oxygen for ATP production. Aerobic glycolysis yields, around 32 ATP per glucose?
Something like that.
Whereas anaerobic glycolysis yields only 2 ATP. It's vastly insufficient for the brain's high demands.
So it needs that oxygen constantly.
Constantly. Consider hypoglycemic encephalopathy. This can result from conditions like certain malignancies that consume glucose or chronic alcoholism impairing glucose regulation. Early protective symptoms like sweating and anxiety rapidly give way to confusion, seizures, coma, and irreversible brain damage if glucose isn't restored quickly.
And it happens fast.
Very fast. A key insight is that symptoms arise long before a global ATP deficit hits the whole brain. As glucose drops, the synthesis of crucial neurotransmitters, glycine, glutamate, GABA, aspartate, is impaired first because their precursors come from glucose metabolism. This leads to severe dysfunction even with some ATP still around.
So the communication breaks down before the power goes out completely.
A good way to put it. If glucose levels plummet further, a devastating process called glutamate excitotoxicity takes over. With depleted energy, the reuptake pumps that normally clear glutamate from the synapse fail.
Because they need ATP to run.
Exactly. Glutamate builds up, overstimulating receptors like the NMDA receptor, causing a lethal influx of calcium ions into neurons. This calcium overload triggers pathways leading to cell death. It's a major mechanism of damage in stroke and hypoglycemia.
Wow. Okay, what about lack of oxygen, hypoxia?
Hypoxic encephalopathy. This results from insufficient oxygen, whether from high altitude, severe anemia, impaired lung function, or poisons like cyanide that block oxygen utilization. Even mild hypoxia causes severe cognitive dysfunction because it impairs neurotransmitter synthesis.
Again, hitting neurotransmitters first.
Often, yes. The brain tries to compensate by increasing blood flow and accelerating anaerobic glycolysis, but this produces lactate and dangerously drops the pH, causing acidosis. Acetylcholine synthesis is acutely sensitive to hypoxia because it depends on acetyl-CoA from pyruvate dehydrogenase, which needs oxygen indirectly.
Okay.
The synthesis of glutamate and GABA is also hit due to the inhibitory effects of elevated NADH from anaerobic metabolism on the TCA cycle in the absence of oxygen. Even catecholamine synthesis is directly affected as those hydroxylase reactions we talked about require molecular oxygen.
So oxygen lack hits energy production and neurotransmitter synthesis from multiple angles.
It's a double whammy, really.
Yeah.
It's also worth highlighting anaplerotic pathways here.
Anaplerotic, meaning filling up.
Exactly. When acetylglutarate is drawn from the TCA cycle for glutamate synthesis, it must be replenished to keep the cycle spinning and producing energy. Two main reactions do this. One uses pyruvate carboxylase, and the other involves the degradation of branched-chain amino acids like valine and isoleucine, which contribute succinyl-CoA back into the cycle.
Amino acids helping refill the energy cycle.
Yes, and this latter pathway critically requires vitamin B12 via the enzyme methylmalonyl-CoA mutase. This just underscores, again, the widespread impact of vitamin deficiencies, like B12 deficiency, on sustained brain function and energy metabolism.
That's a sobering look at how vulnerable the brain is to fuel and oxygen deprivation. Now let's switch our focus slightly to lipids. How does the brain manage its fats, especially for something as critical as myelin, that insulation we talked about earlier?
Right, brain lipid metabolism has some truly unique characteristics. The BBB, again, plays a role significantly restricting the entry of most non-essential fatty acids, so the brain largely synthesizes its own lipids.
It makes its own fats.
A lot of them, yes, including cholesterol, fatty acids, sphingolipids, glycosphingolipids, and cerebrosides. Essential fatty acids like omega-3s and omega-6s are the exception. They are actively transported across the BBB.
Right.
There's also a rapid turnover of lipids, especially at the synaptic membrane, constantly being remodeled and renewed. And crucially, very long-chain fatty acids, VLCFAs, are synthesized in the brain, and they play a major structural role in myelin.
Very long chains, longer than typical dietary fats.
Yes, C22, C24, even longer. And peroxisomal oxidation is vital here, as it's the primary way these VLCFAs and also phytanic acid from our diet found in dairy and some meats are broken down. Disorders affecting peroxisome function, like Refsum disease or adrenal leukodystrophy, lead to accumulation of these lipids and can severely impact brain cells, particularly myelin.
So peroxisomes are key for handling these special fats.
Absolutely. Similarly, lysosomal diseases, where the degradation of glycosphingolipids is impaired due to missing enzymes, lead to their accumulation in brain cell lysosomes, causing various forms of neurological dysfunction, like Tay-Sachs disease. These are stark reminders that lipid metabolism is just as critical as glucose and oxygen for healthy brain function.
So what exactly is myelin then? And why is this lipid-rich structure so incredibly important for our nerves?
Myelin is a truly remarkable multi-layered lipid and protein structure. It's essentially compacted layers of the plasma membrane of glial cells wrapped tightly around axons. Its main job is insulation, enabling incredibly rapid nerve conduction called saltatory conduction, where the signal jumps between gaps in the myelin.
Made by different cells in CNS versus PNS, right?
Correct. In the PNS, Schwann cells wrap themselves around a single axon multiple times. In the CNS, oligodendrocytes are the myelin architects, but as we mentioned, they can extend processes to myelinate segments of up to 40 different axons. Much more widespread coverage.
And the composition is key. You said high lipid content.
Very high lipid to protein ratio, much higher than most cell membranes. It's particularly rich in specific lipids. Sphingolipids, especially cerebrosides like galactosylcerebroside and cholesterol. These specific lipids, along with those very long-chain fatty acids, allow for an incredibly tight, compact packing, which is crucial for its insulating properties.
And proteins holds together.
Exactly. Specific proteins act like glue or structural elements. In the CNS, the major ones are proteolipid protein, PLP, which is very hydrophobic and helps stabilize the structure by interacting with lipids, and myelin basic proteins, MBPs, which are more soluble and found on the cytoplasmic side, thought to bind the membrane layers together.
Okay, PLP and MBP in the CNS. What about the PNS?
In the PNS, the major protein is P0, a large glycoprotein that spans the membrane, and there's also a PNS-specific MBP, known as P2. The precise organization and interaction of these specific lipids and proteins are what allow for such efficient and rapid signal transmission along nerves.
It sounds absolutely critical. So what happens when this finely tuned lipid-rich myelin structure is damaged or its formation is disrupted?
When myelin is damaged, it leads to a class of debilitating conditions known as demyelinating diseases. The most well-known is probably multiple sclerosis, or MS.
Yeah, MS, right.
MS is a progressive CNS demyelinating disease. It's widely believed to be autoimmune, where the body's immune system mistakenly attacks components of the myelin sheath or the oligodendrocytes themselves. This leads to inflammation and myelin loss in patches throughout the brain and spinal cord.
And that slows down nerve signals.
Drastically slows or even blocks them, causing a huge range of neurological symptoms depending on where the damage occurs. MS often presents with relapsing remitting phases, where the CNS actually attempts remyelination, showing some repair capacity.
So it tries to fix itself.
It does try, especially early on. But over time, this repair mechanism often fails or becomes overwhelmed, leading to accumulation of permanent neurological damage. Current treatments primarily focus on modulating the immune system, trying to halt this destructive process.
Are there other types of demyelinating diseases?
Yes, there are also rarer, often inherited demyelinating disorders that stem from specific genetic mutations in the myelin proteins themselves. For example, mutations in P0, the major PNS myelin
The
protein.
one in peripheral nerves.
Right. Those mutations can lead to forms of Charcot-Marie-Tooth polyneuropathy, affecting peripheral nerve function. Similarly, mutations in proteolipid protein, PLP, a key CNS myelin protein.
The CNS one.
Correct. Those are linked to conditions like Pelizaeus-Merzbacher disease and certain forms of X-linked spastic paraplegia. These inherited disorders highlight just how fundamental these specific lipid protein structures are and how their precise formation and maintenance are absolutely non-negotiable for proper nervous system function. Damage the structure, you damage the function. So, to sort of recap our deep dive today, the nervous system is just a marvel of biological engineering, isn't it? Relying on an incredibly diverse and specialized set of cells working in concert.
The neurons and all those glial types.
Exactly. It's meticulously protected by the sophisticated blood-brain barrier, which precisely controls molecular traffic. The brain's communication hinges on a wide array of neurotransmitters, catecholamines, serotonin, ASHI, glutamate, GABA, and others, each with its own intricate metabolic pathway, often deeply reliant on key vitamins and cofactors like B6, B12, folate, thiamine, vitamin C.
All those connections we saw.
And underpinning all of this is a constant high demand for glucose and oxygen to generate the ATP needed for everything from neurotransmitter synthesis and release to maintaining ion gradients. Finally, specialized lipid structures, particularly the myelin sheath formed by oligodendrocytes in Schwann cells, are absolutely essential for rapid signal transmission, and their disruption leads to these debilitating demyelinating diseases.
It's truly remarkable to consider the sheer number of biochemical reactions happening in your brain every single second, just keeping everything running so smoothly. The delicate balance and really the profound interdependence of all these pathways are nothing short of astounding.
It really makes you appreciate the complexity.
It does. And it leaves us with a final thought for you, our listeners, to ponder. Considering how much we've uncovered about the brain's complex metabolism today, what might be the next breakthrough in understanding how even subtle influences, things like diet quality, sleep patterns, maybe chronic stress, or environmental factors, might subtly shift these intricate biochemical networks over time?
That's a great question. How do those long-term inputs affect neurotransmitter balance or barrier integrity or energy production?
Exactly. And how could that knowledge lead to perhaps revolutionary new strategies for supporting brain health, enhancing cognitive function, or even treating neurological conditions in ways that we can only imagine right now?
Food for thought, definitely.
Thank you so much for joining us on this deep dive into the metabolism of the nervous system.
Yes, thank you for your engagement and your curiosity. We hope this was helpful.